We can see you properly.
Yes, let me be able to share my screen. Okay.
So
You
let's
want to
start.
share?
Let's start, sir.
Okay, can I start?
Yeah, just wait for a moment, sir. I just wanted to give a brief introduction about today's program. Good afternoon to all, one and all. I am Dr. Shubha Dwivedi, Associate Professor, Department of Biotechnology, School of Life Science and Technology, Meerut, IIMT University, Meerut. Welcome you all in today's International Popular Lecture. It gives immense pleasure to communicate that IIC-IIMT University, along with Research and Development Cell of IIMT University, is going to organize popular lecture under the scheme of DBT-CTEP, Department of Biotechnology, Government of India, on the topic Next Generation Biotechnological Techniques, a sign QAnon for the ever-increasing global population, with the objective of popularization of biotechnology and to bring general awareness among mass about the basic concepts, recent advances, and potential modern biology and biotechnology. The IIMT University has been established under Uttar Pradesh Act number 32 of 2016. It is the best private university of North India and among top 10 universities in Uttar Pradesh. The university has a rich legacy in the field of education and exemplary recognition in corporate, academia, and public at large. We are committed to provide a world-class education. In addition, the university inculcates an environment of innovation, research, and development coupled with values so that its student becomes tools for socio-economic changes. I welcome and pay my regards to Honorable Chancellor Sir of IIMT University, Shri Yogesh Mohanji Gupta ji. He is a man of vision and under his esteemed leadership, the university is setting milestones. His vision consists to promote Indian culture and research-based education to the new generation. Ever-changing technology and proliferation of new ideas pose new challenges to the universities around the world to develop and deliver innovative curriculum to stay relevant. I welcome Professor H.S. Singh, Vice Chancellor of IIMT University. He is man of research in life science with more than 9,000 citations. His vision clearly states that at IIMT University, we believe in the saying knowledge is power. Our main objective has been developing the young learners on the solid foundations of ethical and moral values, while polishing their leadership qualities, research culture, and innovative skills. I welcome Professor Deepa Sharma, Madam, Pro-Vice Chancellor of IIMT University. She is the guiding principle behind all the research innovation-based activities of IIMT University. How to go for a startup, how learning turns into research, how you become an entrepreneur. Heading towards these teaching areas are her primary knowledge outcome in IIMT University. I welcome all the directors, deans of IIMT University in today's International Popular Talk. Today's eminent speaker is Professor Charles Olivesian Adetunji, Distinguished and Visiting Professor, Center of Biotechnology, Precious Cornerstone University, Ibadan, and Additional Director, Intellectual Property and Technology Transfer, and Head of Microbiology, Edo State University, Uzare. I welcome you, erudite Professor at IIMT University. I just want to read a brief profile of Professor Charles. Professor Charles Olivesian Adetunji is presently a faculty member at the Microbiology Department, Faculty of Sciences, Edo State University, Azare, Nigeria, where he utilizes the application of biological techniques and microbial bioprocesses for the actualization of sustainable development goals and agrarian revolution through quality teaching, research, and community development. He is currently the Acting Director of Intellectual Property and Technology Transfer, Sub-Dean for Faculty of Science, and the Head of Department of Microbiology at Edo State University, Nigeria. He is a visiting professor and the executive director for the Center of Biotechnology, Precious Cornerstone University, Ibadan. He has won several scientific awards and grants from renowned academic bodies like Council of Scientific and Industrial Research, CSIR India, Department of Biotechnology, Government of India, the World Academy of Science, TWAS, Italy, Netherlands Fellowship Program, NPF Netherlands, the Agency for International Development Corporation, Israel, Royal Academy of Engineering, UK, among many others. He has published many scientific journal articles and conference proceedings in reputed international and national journals with over 310 manuscripts. He was ranked among the top 500 prolific authors in Nigeria between 2019 till date by SCI and Scopus. His research interests include microbiology, biotechnology, post-harvest management, and nanotechnology. He was recently appointed as a president and chairman governing council of Nigerian Bioinformatics and Genomics Network Society. He is presently a series editor with Taylor and Francis, USA, editing several textbooks on agricultural biotechnology, nanotechnology, pharma foods, and environmental sciences. He is an editorial board member of many international journals and served as a reviewer to many double-blind peer-reviewed journals like Elsewhere, Springer, Taylor and Francis, Wiley, PLOS1, Nature, American Chemistry Society, Bantam Science Publishers, etc. He is a member of many scientific and professionals, including bodies like American Society for Microbiology, Biotechnology Society of Nigeria, Nigerian Society for Microbiology, and is presently the General Executive Secretary of Nigerian Young Academy. He has won a lot of international recognition and also acted as a keynote speaker, delivering invited talk, position papers at various universities, research institutes, and several centers of excellence, which span across several continents of the globe. He has, over the last 15 years, built strong working collaborations with reputable research groups in numerous and leading universities across the globe. He is the convener for Recent Advances in Biotechnology, which is an annual international conference where renowned microbiologists and biotechnologists come together to share their latest discoveries. He is a president and founder of the Nigerian Post-Harvest and Food Biotechnology Society. Now it is the time. With this brief, now I would like to invite erudite Professor Charles for his talk. Professor Charles, over to you.
I think ma'am, sir is disconnected due to some reasons. We have to wait for a few minutes. Yes, sir is reconnected.
Yes. Yes, Professor Charles.
Yeah.
Yeah.
Okay. Thank you very much. I want to appreciate our amiable. Can you enable me to share my screen?
Not now, sir.
Okay.
I'll share it once again.
Now you are visible, sir. You may share if you want to share your screen, sir. Yeah.
Okay, the host disabled participants from sharing. The host disabled the participant from sharing. Can you enable me to share my screen?
Yes, sir. You can share your screen, sir.
He's saying host disabled participant from sharing screen.
Okay, okay. We are just asking, sir. Vaibhav, sir. Vaibhav, sir. Am I
Hello?
audible? So we are trying, sir. We are trying, sir. Okay. Ma'am, he was getting the difficulty to share the screen. Now I have talked regarding it with Vaibhav sir, and we have permitted. Right now, if sir is connected, easily he can share.
Okay, sir.
Yes, sir. Charles, sir, now you may share your screen because we have permitted from this place. Am I audible, Charles, sir? Yes, sir. Now you are permitted with. You may share your screen. Yes.
Okay, thank you very much. I want to sincerely appreciate the Vice Chancellor of this Meerut University IIMT, and the management, the Pro Chancellor, and the Center for Innovation for inviting me to come as a keynote speaker today. I want to thank Professor Subha Diwani and the husband. I'm very grateful for this great privilege. So this morning, I'm going to be talking about next generation biotechnology techniques. No, the whole world is going through a lot of challenges now in the era of COVID-19 and pandemic. Now we are talking about another strains. So we don't know when all this will start. So that means there is a need for, in order to achieve a stable economy in several parts of the world. I am Charles Oluwaseun. I'm the Acting Director of Intellectual Property and the Distinguished Professor of Biotechnology at Precious Cornerstone University. Also, I'm the Senior Coordinator of Microbiology Society of Nigeria. First of all, before we could achieve anything good, even talking about biotechnology, we must recognize that God is the center of everything, which forms the building block, CDT of our life. Now, it has been stipulated by WHO that the global population is going to increase drastically to 9 billion in the year 2050. If you look at the projection on this screen, you will see from 17th century, in the year 1700, now we are in the year 2021. We have, you can see the population. Now we are projecting year 2050, you can see the population, which means at year 2050, the population is going to increase more than 9 billion. Now, which shows the whole globe just needs to do something in order to feed the ever increasing population. Now, there are a lot of things we need to answer. How do we use all the problem? How do we achieve sustainable city? How do we achieve sustainable food, water and ocean? How does transformation ensure education, gender and inequality? How do we achieve a balanced health and a well-being of mankind? How do we achieve energy decarbonization and sustainable industry? Now, that means circularity and decoupling have come to stay. And which shows innovation, sustainable environments, sustainable agriculture, circular economy is the way forward. Circular economy is a way of living in harmony with ecological system and restoring them. So to do so, there is a need to ensure a kind of life cycle analysis tool, nano-digital empowering, biotechnology, most especially, and also Industry 4 through the use of 3R like reuse, reduce, and recycle. Therefore, there is an urgent need to change to circularity and sustainability, which will ensure better appreciation, that we can embrace a lot of things and ensuring that our economy, our education curriculum is well captured to resolve this. That means we need to drive this accurately. Now, I will say that that shows circular economy encompasses a lot of things. But because of this lecture, I might not be able to pick them one by one. Now, we know Millennium Development Goal has been a kind of impetus towards sustainable development goal. So if we are to resolve the global challenges, global problem, we need to improve on Millennium Development Goal and embrace sustainable development goal. Now, this is showing various aspects that we can tailor that has formed a kind of innovative we're going to use to solve global problem. Now, we as a biotechnologists, what is the model we can adapt to solve global problem? And how do we define sustainable development in terms of real project in village and in real life? That means we need to go back to our community. We need to identify the problem there. We need to understand the challenges. We have to emphasize, we have to define, we have to ideate, and build a representative of idea, a prototype. And now we have to go back to the community to resolve that problem. Now, the whole globe is going through a lot of food insecurity, social insecurity, a lot of governance problem, education, gender inequalities, and a lot of things. Now, there is a big question before us. Can biotechnology be a sustainable tool that could help to resolve the global problem, and how will we achieve sustainable development goal? Now, I want to share some of my personal experience in the area of biotechnology, which are based on sustainable development goals that form a kind of bedrock that are going to solve the global problem. Now, as I said before, that the world population is going to reach 9 billion in the year 2050, and HEPO have stimulated this. If we are to support the ever-increasing population, that means agriculture needs to undergo a drastic transformation in order to feed the ever-increasing population. Now, there are 4 major goals of our time. How do we achieve food security? Almost 870 million are hungry presently. How do we avoid dangerous climate changes? What are the roles environmental, agricultural sector, forestry sector? Health sectors have a lot of roles to play, especially in this pandemic season. Now, we graduate a lot of students from our university, research institute and everywhere. How do we secure the garbage on? So these are the things. Now, there are 4 major problems that can help us to solve our problem of food insecurity. Number one, we need to reduce food waste. And especially one of the food products is produced from one farm to home. One of the solutions is to upcycle the food that will have been thrown away. Also, government need to change laws that unsold or unable food must be donated to food bank and soap kitchen. Number two, that can help us to achieve food security is that we need to shift diet to more plant-based. In India, a larger percentage of population consume a lot of plant-based food. And when you check, many people are very healthy, which shows that a lot of high diets that are rich in meat are almost wasteful. Meat from cow, sheep, goats uses precious resources, and demand meat and dairy products is steadily growing. So today we have more meat substitutes available in the market that are made from plants, and we need to embrace a kind of vegetarian method. Now, we need, because we can achieve our food security, we need to boost crop production. So we as a biotechnologist, we need to increase crop production without increasing the amount of land use. Now, land is becoming a problem in some cities. We need to change and embrace a lot of technology like hydroponic farming, because the whole world is increasing daily and we need to feed the ever hungry world. The amount of crop harvested needs to be increased from 1 year to another. Now, one important biotechnological techniques is CRISPR technology or gene editing could help the plant breeding to desire a particular trait, a drought, which could avoid a lot of resistance and subjectivity to disease and past. So it is not the same as GMO, which misses gene to 2 or more species. Another way to boost less land with sustainability is a lot of indoor vertical farming. So we need to embrace it. Another ways we can achieve a food security is to look into the sea for more food. Now, with the head population growing, turning to the world ocean for food, we need to embrace new blue food revolution. That is, taking more fish out of our sea and inland is not the answer. So we need to look into this. Increasing agriculture, indoor fishing farm and ocean farm is going to help in this regard. Another way we can improve food security is using an agroecological approach to food production. Now, it will be stated by UN Committee on Food Security, which has draws an agricultural approach that incorporates sustainable agricultural food system. Now, agroecology manages nature, replaces things like chemical fertilizer, pesticides, insecticides. So combination of plant raising can increase land production. Now, this is a very similar way to permaculture, that's a movement that uses natural way to build a kind of natural soil and regenerate agriculture. So you can see there are a lot of techniques around us that we need to do. Now, what is the way around? What are we saying that's about technology? We have come to realize that we need a sustainable agriculture, which involves the practice of farming using principle of ecology and sustainable relationship between organism and the environment. Now, sustainable agriculture encompasses around people, planet, and profit. Now, around the world, production of crops and goods is not our problem. We have the techniques, we have a lot of practice like good agricultural practices, HACCP, SOP, GMP, and a lot of all the techniques are there. But how do we resolve the issue of post-service? Production is not our problem around the globe, but how do we save? How do we ensure we don't have waste? That is different between waste and food loss. So we need to evaluate this. So during some of my works, I am a research cluster of a new herbicide that can assist farmers to refine the usage of synthetic herbicides, which have become a threat to food safety, ecosystem, and human health. Now, this is showing some of the procedure we followed during the experiments. We have established the molecular characterization of the isolates. This is showing the sequencing of the isolate, the blasting elements, and this is showing the homology that shows our isolates with various isolates that is producing this bioavailable strengths. We have documented this in an high-impact journal, and we have also established the active ingredient responsible for biological activity and herbicide activity of this particular strain. We went further to establish how these isolate performance works, and we established a monitoring using non-target rights and scanning electron microscopy and transmission electron microscopy using the phytotoxic metabolites from this active strain of the celestial wave. We have also established the biochemical property physiological activity and the socialized change on the tested weight, which has been documented, and we have published that in a high impact factor. So this is showing the effect of the biopsy strains on the tested weights. Now, after we have established that, we move further to establish the main biological activity. So we went further to establish this using the metabolomics action. Now, during this, we scale up from small level to a higher level using different fermenter. And we have established, and this is showing the various biological activity responsible for the biological biobicide activity from our strains. So we have used techniques like HPs, LC, MLs, APRs, MESLs, neutron organoresonance, protein NMR and carbon NMR. We went further to show the carbon possibility, which was determined by the DET, the Sotolas enhancement polarization transfer, where established the 45, 90, and also COC using bronchial microgram. So this is showing the paper where we have established the biological activity of this particular strain. We went further to show maybe where these biobicide are economically feasible, most especially how do we mass produce them for farmers that cannot speak English. So we came about a model, Animation Without Borders, they could help the farmers, and we went forward and we have shown that the synergetic effect of a raminolipid will enhance the activity of the biobicide strains and also could reduce the level of toxicity that normally experienced in toxic herbicides. So this is showing the process involved in the production of the metabolic product from these strains. So we have established the journal entries. We went further to establish the use of available carbon sources because microorganisms do depend on nitrogen cells and chemical cells. So we look around and experiment and validate the common grains using sorghum. So we produce and mass produce this on sorghum, and we gave it to farmer because farmer it's easy for them to use this when compared to synthetic media that is very costly. So they could be able to produce it. So whether can it be a kind of eco-friendly herbicide? So we have established the ecosocial assessment of this herbicide, and we established the biocompatibility by showing the extraction of the soil of the microorganism, the cycling of the essential soil nutrients, and we have also established all the soil biocompatibility like carbon evolution, organic carbon content as activity, and essential microorganism activity, how they interact. And we have established that it's eco-friendly and is working. And we have established this also in an input practice garden. We went further to show how can we demonstrate this in a practical approach on the farm. And this is showing where we have used. And our research shows that a treatment for exhibits the best biological activity. Now, in terms of costs, we went for can it be comparable to highly costly already established herbicides, chemical synthetic herbicide, and we show our study shows that it's more cheaper when compared to chemical herbicide. Not only that, we went further to file a patent because innovation has become the key answer to global challenges. So we as a microbiologist, I want to encourage us. So this has been approved by the federal government of Nigeria in this regard. Now, during COVID-19, the whole globe encountered a lot of problems. So we came up as a scientist that how can we resolve the global problem? So we embraced the use of bioinformatics tools. This is a collaboration work with Dr. Wadazani from India, in Delhi, and a lot of other scientists in the group. We have a bioinformatics research group. Dr. Wadazani is a group leader. What do we do? We try to establish evolution and protection of African cassava mosaic carbon virus called protein. And we established that cassava is a very important stable food that is cultivated widely. African contributes almost 61% of the global production of cassava. It serves as a kind of stable food that is used. I have a lot of wide application, whether industry and food industry. They make use of a lot of it, and a lot of family consume cassava every day. Now, but one of the major challenges, as I'm talking about post-harvest before, is cassava mosaic disease caused by African cassava mosaic. Now, the yield worth in US ranges around 1.9 to 2.9 billion per annum. Now, we have discovered that the coat protein of this virus has a kind of multi-interactive role that could help and serve as a kind of biopesticide against this virus. And this, there have been a lot of that information, whether ACM, Biotechnology, or the protein could work as about herbicide or about pesticide. Now, could this form a basis to announce the effective management of this virus? We went further to use this, so we established a lot of bioinformatics and biotechnological tools to establish this, and we have done this. This is showing a various code content of the family. This is showing the homology of these various code protein, and this is showing the cassava mosaic virus. It's showing the alignment, and this is showing distribution of the concern motif, and this is showing the three-dimension homology of the various, and this represents the biological process ontology, the cellular component ontology, the molecular function ontology of this particular thing. So what do we discover in the registry? We discovered that SCMG component belongs to nuclear export factor of the family of germany called protein, and the phonogenetic analysis shows existence of a very strong affinity among the African isolates, and MSA gene with very high concept domain and three motif and gene analysis precise, and this is showing the publication from this particular one. We went for that to another project. How do we establish the application of cyclopin P450 in agriculture, in molecule, in industry, in food industry, and in environmental biodegradation? As we have a lot of xenobiotics and a lot of compounds that are toxic. So how do we understand the application of cyclopin P450 in maintaining a cleaner environment and in food security? So we established a lot of work in this dimension, and a very renowned scholar, scientist, Dr. Wadazane Palami, won a grant for this particular project by the American Society of Microbiology and Federation of European Microbiology Society. And we have presented the work which was sponsored by Nagar, and a lot of work have been documented in this regard. And so we use CRISPR a lot in our research group. And as you know, this year Nobel World Prize was won on CRISPR. Yeah, CRISPR sequences are derived from DNA sequence of bacteriophage, previously inverted prokaryotes. So in our research group, we went further through a collaborative project from USA, European, African, Asia, Pacific, Caribbean Nano Initiative. We discovered that farmers encountered a lot of problem in order to pass their biopesticides in the crop. So what do? We try to define a kind of nanotechnology that embraces biological material and ensure easy delivery and targeted delivery to crops. As you know, the cell walls of plants are surrounded by a very hard cortical of the cell wall. So these nano have a kind of high effective delivery that will ensure our delivery. So this is showing the grants we have won in this regard. Now, we went further to see, could CRISPR help in a lot of eggs, and we have established that. No, not for that, we went further to see how we could produce biosurfactants. Could it be a permanent replacement to costly environmental and chemicals? Can we use it in management of a lot of medical significant pathogen? So this is showing the metabolomic phase of the work. We went further to establish the proteomics aspect. We went further to establish the genomic aspect of the work and the bioinformatics store to implement. This showing the optimization and the standardization of the biostandardization of the biosurfactant that will help us to antibiotic. We have went further to establish it and publish it in a high-impact journal. We went further to see who is every farmer on the farm. So this is showing the journal that we use in establishing this farm. We went further to establish the enzyme activity to work to enhance biodegradation of leaf. So you can see the difference between the raminolipid twitter leaf and the control that was not prepared. Now, we went further to document the patents in this regard. Now, we went further to see how can we help farmers, most especially the animal farmers that are going through a lot of challenges How can we discover? So we went through in our research group to discover a simple biotechnological techniques that could help them to solve their problem. As you know, that many farmers take recommendations need to produce their own animal feed. And a lot of world are going through a lot of agricultural waste that has been discharged into the environment, which constitute a lot of anthropogenic activity. Many of these wastes are generated in turn, and they now become a kind of economic and ecological challenges. So in our previous work, we have used solid state fermentation, biophasic fermentation, and submerged fermentation. But in this context, we have established the use of solid state fermentation to produce animal feed, and it's very nutritious. And we discovered that the mutant strains also behaved a very wide activity when compared to the strain. We went further to see how does it work when we use this to feed chicken, and we established a very good result, and it's very potent and very active, and it gives farmers a lot of gain. We went further. All these agricultural waste, can we turn them to high relevant, biostable product? So we have established using biotechnology to establish this, and it has been published. Now we went further. I was talking about the issue of post-storage the other time. Now, during some of my work, I went further to establish how biological activity of active compound from natural compounds could extend the shelf lives of vegetables and crops. So we discovered a lot of edible coffee and edible things. Now, this is showing the process in processing center. We have a control modified atmosphere and more refrigerators and a lot of other storage techniques. So what we use, and it's a collaboration project with a lot of universities like Ibu University and Nigeria Water Research Institute. We have done a lot of work extensively when I was there as a principal scientist. So we went further to see, can farmer cope with this costly equipment? No, definitely we came up with a very simple material that will enhance farmer to store their produce. So we went further to see from waste, you can see crab waste, we turn it to chitin through deactivation, deproteination, and a lot of other processes. We have been able to form a new edible coating that will exchange the shelf life of fruit and vegetable. We have established this, and these have also been published in a high impact journal. We went further to see how criticism will extend pepper, chili, and a lot of other crops. Also, these have been documented. Many people suffer from diabetes and sugar-related diseases. Our group came up with the glucose syrups that could be used to manage the issue of sugar. We went further in part of our research, which I did in Jawaharlal University in New Delhi, in collaboration with a lot of distinguished scientists. We discovered that endophytes could save people from a lot of food storage. So as a biotechnology and microbiologist, we've discovered that it has a higher growth promoting effect and enhance caused by plant hormone with animal produce, and they could prevent a lot of diseases. We went further to see the activity of nanotechnology when combined with the metabolite produce from many of these endophytic microorganisms. We now discover that they have ability to tolerate diverse environments, they are biodegradable, eco-friendly, and they could be synthesized with a lot of metals. This is showing the metabolic activity and action of this particular strain. So we went further to establish various biological compounds present in many of these endophytic microorganisms. So this is showing a lot of them. We went further to see LCMS to demonstrate the biological activity of the active component responsible for this biological activity, which was done at ARF at Delhi. We have established, we went further to synthesize the nano, and this is showing the nano synthesized and we could extract. So we have done the optimization of this story, and we went further to use transmission electron microscopy, high resolution microscopy, and diffraction, electron diffraction, and a lot of EVR, zeta potential to establish the nanocompound that has been synthesized. And we have established this. We have used scanning electron microscopy to also examine the plates. We have used atomic microscopy, also pulse microscopy, to establish the size, the topography, and the structure of the nanocapillary. With this step, we went further to establish the parts and the biophysical activity of these pesticides, and we use zeta potential and dynamic light scattering to establish these facts. We went further to see will this be stable after a long period of time, because many scientists encountered a lot of instability in the use of nanoparticle, and we have used X-ray diffraction to establish this. As I was talking about post-harvest, a lot of disease called this, so we use this newly synthesized nanoparticle in a biological activity. We have established the analogy activity, we have shown the biological activity of this isolate, and we have established the mode of action. We have also shown that it can be applied in the environment. We have tested various parameters, the acidic phosphides, alkaline phosphide, dehydrogenativity, catalase, and a lot of other stuff. We have also shown that it ensures some of the isolate that ensure cycling of essential nutrients like nitrogen, carbon, in the soil. So also we established in the greenhouse that is working, and we went further to see could endophytes be used to ensure adequate earth delivery. So we use nanotechnology, biotechnology, and bioinformatics in this regard, and we have ensured that some of these isolates are working. They can ensure that we achieve food security, and these are showing some of the results we got from our work. Now, this is some of the published work in this regard. Not only that, I want to encourage all of us, we need to develop a robust collaboration with agricultural extensionists. Now, we have many of these technology, we have many of these innovations, but we need to carry farmers along. We need to conduct rallying rural support, mostly at the hotspots agricultural community, which are in the rural environment. In order to show them the best biotechnological approach they could adopt. We went further to establish a patent and file a patent in this regard. And I want to talk about some of our work in the area of environmental biotechnology. Now we have used techniques like denaturing relevant gel electrophoresis, amplified ribosome DNA restriction analysis, terminal restoration, terminal length morphosis, single-stranded conformation, and a lot of them. Now, you know the whole world is going to issue of coronavirus. Now, we as a biotechnologist, academicians, students, data scientists, needs to come up and exploit the use of artificial intelligence, machine learning, in order to solve this global problem. Artificial intelligence has helped us in fighting this dangerous disease very early. It has given us a kind of emergency response, critical decision-making surveillance. But we need to embrace on this. That means we as a biotechnology need to develop a robust research development blueprints which need to be activated in order to accelerate diagnosis, vaccines, and therapeutics. And this blueprint is going to improve coordination between scientists and global health professionals and accelerate the research development process. And we need to develop norms and standards in this regard. So in this regard, I am a research group performing in silico screening of medicinal plants, potentially a bit of transferrin protein, serine, applied by NC-SARS into human cell. And we have used a lot of techniques like drugs like nanoparticle to establish this by in a kind of in silico bombing. And the research shows 3 phytochemical active compounds that are present in moringa, Singingia ovalis, and Anona, which are niserine, cotyledon, and morphine. So we have used a lot of these techniques to establish these facts. And this is showing some of our results that we got. This is showing the PROC chart Rackman, and this is showing the model we got from these results. This is showing the 3D kind of structures of the popping. This is showing the interaction between the residue. This is showing another interaction, and the paper has been published in an high-impact journal. And most of our work, we have used CRISPR-Cas system to treat several diseases and other diseases. And as I've said, among chief advances in post-regular intellectual references, CRISPR has become a novel tool that can help us to edit a lot of gene and has multiple applications. So it's a kind of revolutionary tool for gene editing and kudos to the great scientists that helps in this discovery. And also, many scientists have used these particular peptides to resolve the global problem we are currently facing, coronavirus disease. And it has become a kind of tool because it has a low cost, rapid detection, and it's as well a kind of development of a one-pot as a platform. So this has helped a lot of scientists to talk about a lot of results. Now, during this pandemic disease, we have consulted for a lot of farms, a lot of hospitals. We have done a lot of awareness, sensitization, and we have come up with a kind of biomedical technology and health informatics robust project. It's a collaborative project with Professor Fatima at London School of Hygiene in UK. We have established the use of machine learning management of COVID, artificial intelligence, deep learning, extended real learning hub, virtual augmented information resident, and we have done a lot of IT-driven robotic and their role in health care. Cybersecurity issued the 3 application of 3D and a lot of them. Now, a lot of scientists have established CRISPR, how it could be used to manage sickle cell analysis, how it could be used in gene therapy and sickle cell anemia, and particularly they use a lot of drastic diseases. Now, researchers have used CRISPR to solve a lot of liver diseases, and also can this have been used to eliminate issue of global shortage of organ. Now, many scientists have established how it could be used in resolving malaria problem. They have used it to solve diverse reproductive problem. And a lot of researches have been done on CRISPR berries, gene therapy, and a non-variable human embryo. Now, a lot of scientists have established the application of CRISPR in managing HIV disease. It has been used in fixing cancer disease. It has been used in fixing a lot of bad blood clinical hematological challenges. So you can agree with me that hematological diseases are very common. So the use of CRISPR will help in edit hepatitis site to understand the model of blood disease and develop a kind of novel therapy for a lot of hematologic malignancies. Now, CRISPR-Cas have been used for medical genetic screen application, and a lot of perspective has been demonstrated in this regards. Now, we need to embrace a lot of bioresources. We as a biotechnology, we have a lot of bioresources around us. How do we not create resilience through the effective bioresource creation, expansion of products and bio industry? So all of us as a scientist, as a stakeholder, as a center of SNL like IIMT University, we have been doing this. We are so proud of what you are doing. So the whole world needs to learn from you. Edo University is also doing great in this regard, and many great universities, Precious Cornerstone is doing great in this regard. So sectors need to work together. Identify bioresources creation, expansion of bioindustries and bioproducts. How do we use this in med sector to improve therapeutic drugs, establish pharmaceutical manufacturing new vaccines, and a lot of things? How do we do them in industry, by plastics, by composites, bulky, by ethanol, by visuals? We need to embrace this. How do we use them in our bioenvironments, in bioabsorption, bioleaching, biomedical, bioremediation of highly polluted water and soil? We need to embrace this. In agriculture, we need to embrace the use of bioresources in production of our fertilizer, animal vaccines, improvements of energy, crop initiative, water management, production of food, agriculture, conservation, and a lot of them. Now, in Africa, we have a lot of available bioresources. We as a biotechnologists and microbiologists have come up with the concept of African fermented dairy product. Now, we have to improve all these, our naturally produced bioresources in order to solve the global problem, because they are very steep areas. When I was at Jawaharlal University in Delhi, I came up with a new innovation. How can we convert waste to waste? We have a lot of waste around us. We have a lot of unrecycled materials. So grafting, bone grafting, have become a kind of medical tools that we can use to resolve a lot of problems that surrounds a lot of bone problem, bone challenges. So autografts, allografts, synthetic biosimilars and swab others regular. Now, I would say that makes also conductive proteins, also conductive matrix, and cytogenic cells. We need bone grafting to solve this problem. So what do I do in this research? I gather a lot of available agricultural waste, like egg waste, rubber waste, in forming a natural bone that could help in fixing a lot of bone-related problem by producing a biogas from this agricultural waste. Now, this is showing some of the results we got during this evaluation. Now, another tools that will help us about technology is nanotechnology. Nanotechnology will help us to ensure a circular economy and ensure a stable environment. So, in most of our work, we have used nanomaterial to establish a lot of facts that nanotechnology has become a tool and parts of biotechnology that will help us to ensure a cleaner environment and a sustainable environment. Now, as a biotechnologist, can food serve as a medicine, a subject food? Yes, we have used a lot of bioactive compound in our group to establish that this could work, this could ensure food security, and there are a lot of them are bioresources around us. We need to embrace them, we need to use them, we need to utilize them. This could help us to solve a lot of problems like cancer problem, prostate problem, kidney problem, and a lot of diverse problems. Now, we have all around us a lot of essential health. So we as a biotechnologists need to look further into how we could use all this in resolving global problem, use of mushroom in resolving global problem. So conclusively, you will bear with me that biotechnology has become a kind of tool that will ensure a circular economy, provide a job opportunity, improve drug, vaccine therapy, most especially in this era of COVID-19, we could maintain a clean environment and to improve animal health and health. Now, before I start my lecture, I want to embrace this that we as a scientist need to work together. Transdisciplinary, interdisciplinary, multidisciplinary, and monodisciplinary. We need to work together. We need to form a great research cluster before we could resolve the global problem. So interdisciplinary have become the order of the day. So I want to thank all of you for this great privilege. It has been very amazing giving me this opportunity to give this year India Scientific Popular Talk. I'm so honored to be invited from Africa to talk. And I want to appreciate many of the countries, Germany, Israel, Canada, most importantly, Indian governments have invited me several times without non-military governments to come as a visiting scholar to a reputable university and share my word of knowledge. Now, I want to invite all of you to join us in next year recent advances in biotechnology. This year, we honored Professor Subal Diwali and the husband, Professor Diwali, also were there as a keynote speaker. They were honored as a distinguished scientist. We look forward to see many of you in next year. This is showing some of the previous picture of the recent advances. You can see our renowned professors, Pandey, Professor Ajit Verma, and many, Professor Paromi, Professor Bob, Professor Yerima, many, Professor Loke, and many of them I could not mention. So we are seeking for a collaboration for next year that we could collaborate and establish recent advances together. It will be our joy to see you. Now, I want to advise all of us also, we should not just have all these technology. We need to liaise with our governments. We need to lobby with the government. It is when we lobby with the government and we put government and policy maker in place, that is when we could solve the problem. I remember in the year 2016, when Nigerian agriculture was banned to Europe, I was one of the consultants that consult for the Senate of the Federal Republic of Nigeria in order to resolve this issue. So we need to relate with the government. On this note, I want to thank many of the great professors that have helped me. And in India is my second country. I want to thank a lot of many of them that have sincerely helped me, Professor Sabu Thomas, Professor Suwadi, Professor Adivama, Professor Achok Pande, Professor Nero, And many of them I could not mention here. And these are some of the great memory from India and around the globe that I have visited during my short stay in India. And India is a very great country. We are so proud of what you are doing, and we are happy. I want to thank many of them, and it's a very sincere pleasure being around in all these. Also, I would like to invite you to take benefit of many editorial advantage that I am a series editor of. I will be willing to have a project collaboration with all of you. It will be my sincere thanks. So on this note, I would like to appreciate our Vice Chancellor, Professor, Distinguished Professor, Yogesh Mogesh Gupta, the Chancellor of IMP. We are so proud of what you are doing, and the whole globe is so proud of you. I want to thank the Vice Chancellor also, Professor Adhika Second Singh. It's a great privilege for me to give this year annual popular scientific talk. I want to thank Professor Diwali and the wife also, Professor Shobha Diwali. Thanks so much. I'm so honored for this great privilege. Thank you. Namaste. Stay in touch and looking forward to collaborating with all of you. Thank you very much.
Thank you so much. Thank you so much, Professor. I just wanted to give a brief note, whatever you discuss over here, that biotechnology has come of age. Is the application of our knowledge of biological processes as our knowledge of the natural world grows. So it is applied to the processing problems of our society like health, food, safety, cleaning environment. Biotechnology's greatest impact remains in healthcare. 40% of the drugs now being tested come in part from biotechnology. Dozens of proteins are used every day as medicines and the product of recombinant DNA technology. Clobusting enzymes, growth hormones, antibodies for cancer, arthritis, tissue transplantation are now part of standard hospital care in India. Biotechnology has started to be applied in other industries. Despite widespread consumer concern in Europe, biotechnology crops now cover an area of land nearly as big as Spain. This is only the beginning. As biotechnology merged with chemistry, physics, in the world of nanotechnology and many more other new sciences, which stated by Professor Charles. So biotechnology is just a boon for the society. And I'm just giving a brief note on whatever Professor Charles stated in today's talk. And I'm really extremely thankful, Professor Charles, for your keynote speech under the umbrella of DBT sponsored program. This is popular lecture series. And it's really very interesting, whatever you have discussed, and definitely it will create a much impact on the society and much impact on the students too. I'm highly thankful for our Chancellor, Sir, Shri Yogesh Mohanji Gupta, who provide the opportunity. I am thankful to our Vice Chancellor, Sir, Professor H.S. Singh. He is also the man of science, man of research. He has more than 9,000 citations. And I'm thankful to Professor Deepa Sharma, Madam Pro, Vice Chancellor of IIMT University. He's women in science. He's very much with the innovative mind. I'm thankful to the team. I'm thankful to the co-coordinator of this event, Mr. Nitin Agrawal. Without his support, this task cannot be comes up to the end. I am thankful to the technical team. I'm thankful to the Vaibhavji for creating all these type of facilities. I'm really thankful, and I am just giving here that kindly share this video, this YouTube link as much as you can. So this is a thankful note from IIMT University, from my side, from Dr. Shubha Dwivedi's side. Thank you so much, sir. Thank you so much.
Thank you, sir. Thank you, ma'am.
Thank you, sir.
Thank you.
Hello, Vaibhav sir. Ma'am, off. Can we off?
Yes, yes, I can hear. So whatever query we received, I can just mail you. So whenever you get the time, you can just answer for those queries also.
Okay, no problem.
Yes.
Okay, sir. Thank you. Thank you very much.